Every day is a beginning. Take a deep breath, smile, and start again. If you are positive, you will see opportunities instead of obstacles. Always remember that your present situation is not your final destination. The best is yet to come. Blessed day, my dear students. I am Chitra, welcoming you all to this online class. We have started the 5th chapter and completed up to RNA world. Today, we will discuss about properties of genetic material, packaging of DNA helix, and DNA replication. We all know that DNA is genetic material. The experiments by Hershey and Chase clearly indicates that it is DNA that act as a genetic material. The properties of genetic material includes self-replication. Second one is the stability. Third one is the information storage. And fourth one is the variation through mutation. So we will discuss the properties of genetic material one by one. First one is the self-replication. The genetic material should be able to self-replicate. So according to the rule of base pairing and complementarity, both nucleic acids, DNA and RNA have the ability to direct duplications. Here, the protein do not duplicate or replicate. Proteins fail to fulfill this criteria. Second one is the stability. The genetic material should be stable structurally and chemically. The genetic material should be stable enough not to change with the different stages of life cycle, age, or with change in physiology of an organism. Stability is one of the property of genetic material because this genetic material should be stable so that it can able to transfer from one generation to the other generation. So what are the evidences for the stability of DNA as genetic material? So this was clearly given by Griffith transforming principle. Griffith transforming principles gives an evidence for a stability of genetic material. Heat, which killed the bacteria, did not destroy some of the properties of genetic material. Heat can kill bacteria, but it can't able to destroy the properties of the genetic material. So, DNA is a stable genetic material. Next, we will move on to the stability compared to DNA with RNA. Here you can see the DNA is a double-stranded whereas RNA is single-stranded. The two strands of DNA are complementary. Because of that, if the strands are separated by heating, they can come together by renaturation. Second one is the 2' carbon OH group present in each nucleotide of RNA is a reactive group. It makes RNA reactive and easily degradable. So this RNA is reactive and easily degradable, and RNA is catalytic and reactive because of 2' carbon in the OH group of present in each nucleotide. Fourth is, compared to RNA, DNA is more stable and less reactive. And presence of thymine confers additional stability to DNA. But instead of thymine, RNA contains uracil. Next is the information storage. The genetic material should be able to express itself in the form of Mendelian characters. RNA can directly code for protein synthesis and can easily express the characters, whereas DNA depends on RNA for synthesis of protein and can express the character. DNA is more stable and stores the genetic information. RNA is less stable and RNA transfers the genetic information. DNA stores, whereas RNA transfers the genetic information. Next is the variation through mutation. The genetical material should be able to mutate. Both DNA and RNA are able to mutate. RNA being unstable mutates at a faster rate. Viruses having RNA genome with shorter lifespan can mutate and evolve faster. So this RNA is unstable. Both RNA and DNA can function as genetic material, whereas DNA is more stable, so it is preferred for the storage of genetic information. So let us have a conclusion or a summary of these properties of genetic material. First one is the self-replication should be able to self-replicate. The genetic material should be stable structurally and chemically, should be stable enough not to change with the different stages of life cycle, age, or change in the physiology of an organism. Third is the information storage. It should be able to express itself in the form of Mendelian characters. And last one is the variation through mutation. The DNA material should able to mutate. This property is essential for evolution of a life. These are the properties of genetic material. So DNA has all these properties. So it is the genetic material. Next, we move to packaging of DNA helix. The length of the DNA double helix in a typical mammalian cell is 2.2 meters. 2.2 meters. The average diameter of this nucleus is 6 micrometer. That is 6 microneutron. So, the length of the DNA helix in a typical mammalian cell is about 2.2 meters and diameter is very less. How is it possible for such the length is occupied inside the small nucleus? There are about 6.6 into 10 to the power of 9 base pairs in a human cell. The human cell is diploid and it consists of 6.6 base pair, that is 6.6 gigabases or 6.6 billion base pairs. We know that the interbase pair distance is 0.34 nanometer. What is interbase pair? The distance between two consecutive base pairs is called the interbase pairs. So with help of this, we should calculate the total length of the double helical DNA. Double helical DNA is equal to total number of base pair into distance between two consecutive base pairs. That is 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of minus 9, that is equal to 2.24 meter. So the total length of the DNA is 2.24 meter. The length of DNA is 2.24 and the nucleus diameter is only 6 micrometer, that is 0.00006 meter. So, how is it possible to have such a long polymer packed inside the cell? How this package of cell takes place? So, there are many theories of packaging the DNA. The first theory is the unanime model Duprat in 1965. So, in this unanime model Duprat, the explains a single-stranded DNA model as a long-coiled molecule which is associated with histone proteins in eukaryotes. This model explains single-stranded DNA model, which is a long-coiled molecule. So, this unitime theory stated that a chromatid represents a single linear DNA molecule and this is a absolute now and this is not used now. So, next is the how do the prokaryote pack inside the DNA? Prokaryotes such as E. coli do not have a well-defined nucleus. So, these prokaryotes do not have well-defined nucleus. The DNA of E. coli is held with some proteins in a region called the nucleoid. So this nucleoid, the DNA is organized into large loops held by proteins. So in a nucleoid, the large loops of proteins are held together inside the prokaryotes. So genophore. What is genophore? Genophore is a special term. The DNA of prokaryotes is almost circular and they lacks the chromatic organization. This is called a genophore. Genophore is nothing but the nucleode contains this genophore. So it lacks chromatin organization. Genophore of prokaryote consists of supercoiled DNA. What is supercoiled? Coiled and coiled. So the coiling is more. Genophore of prokaryotes consists of supercoiled DNA. So genophore is the DNA of prokaryotes. Histones and chromatin are absent in prokaryotes. Histones are absent in prokaryotes and also chromatin is also absent in prokaryotes. We will see histones and chromatin in the next slide. Organization of DNA in eukaryotes. Organization of DNA is much more complex in eukaryotes. Chromatin is formed by a series of repeating units called nucleosomes. There are 5 types of histone proteins. There are 5 types of histone proteins. They hold the DNA in position. How do these proteins hold the DNA? How do this protein hold the DNA? DNA is a negatively charged and proteins are positively charged. So they are held together. What is a chromatin? Chromatin is a complex of DNA and histone protein. Chromatin is a complex of, it's a mixture of DNA and histone proteins. So what are histones? Complex small proteins are called as histones. Complex small proteins are called as histones. So histones and DNA combine together to form chromatin. What is the primary function of chromatin? The primary function of chromatin is packaging the long DNA molecule into more compact, denser structures. So organization of DNA is much more complex and there are 5 types of histone proteins. How many types of histone proteins? There are 5 types of histone proteins. These 5 types of histone proteins are H1, H2A, H2B, H3 and H4. I repeat, H1, H2A, H2B, H3 and H4. These are the 5 types of histone proteins present in eukaryotes. Let us see the structure of nucleosome. Kornberg model. So this model is actually called as Kornberg-Thomas model. H2A, H2B, H3 and H4, these are the 4 histone molecules organized to form an unit of 8 molecules called histone octamer. So what is histone octamer? These 4 histones H2A, H2B, H3 and H4 organized to form a unit of 8 molecules. Octo is nothing but 8. Mer means fusion. So octamer. 2 histone molecules from each type of 4 histones H2A, H2B, H3 and H4 are organized to form a unit of 8 molecules is called histone octamer. So it is a very important 2-mark question. You have to memorize it. So the histone particles are packed together. So how these histone molecules are packed? 2 molecules each from H2A, H2B, H3 and H4 are organized to form the histone octomere. So in this video you can able to see how it is formed. A negatively charged DNA is wrapped around the histone octomere to form a structure called nucleosome. What is called nucleosome? Negatively charged DNA is wrapped over this. How it is wrapping you can able to see in this video. So this nucleosome is nothing but a negative charged DNA is wrapped around the histone octomere to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. It contains how many base pairs? 200 base pair of DNA helix. We can able to see the structure of a nucleosome. So let us see the structure in detail. So this we can see histone octomere made up of H2A, H2B, H3 and H4. Four types of histones are inside the octomere. So you can able to see in this picture. There are four types and the fifth type H1 is outside the octomere. The DNA is wrapped around the octomere. Two histone molecules, each type of the four histones H2A, H2B, H3 and H4 are organized to form an unit of 8 cells that is called histone octomere. The histone octomere are in close contact. The DNA is called coiled on the outside of this nucleosome. The DNA makes two complete turns around the histone octomeres. It makes two complete turns. These two turns are sealed off by this H1 molecule. You can see here. So, the linear DNA is exposed to the enzyme. This linker DNA. The one nucleosome is combined with another nucleosome by a linker DNA. Neighboring nucleosomes are connected with the help of this linker DNA that is exposed to enzymes. These nucleosomes are connected by DNA linker. So, the composition of nucleosome is the histone octomere, histone ions and the DNA which is wrapped around it. So, this nucleosome you can able to see like a beads on a strand. These nucleosomes are connected. There is no histone H1 particles. So, these nucleosomes are connected by the linker DNA. Chromatin lacking H1 has a beads on string appearance. Actually, the nucleosome has H1. So, this chromatin lacks H1 which shows bead appearance. The diameter of the beads of string is 11 nanometer. In this structure, DNA enters and leaves the nucleosome at random places. So, this DNA enter and leaves. So, beads of a strand nucleosome. Here, the link DNA attaches each nucleosome and you can see like in this picture. H1 of one nucleosome can interact with the H1 of the neighboring nucleosome, resulting in further folding of the fiber. So you can able to see in this picture. So one nucleosome interact with the other nucleosomes. So 30 nanometer fibers arises from the folding of nucleosomes. So in this picture you can able to see this the nucleosomes are inside it. The chromatin fiber in interphase nucleus and mitotic chromosomes have a diameter between 200 to 300 nanometer. So the diameter is 200 to 300 nanometer and represents a inactive chromatin. This structure is stabilized by interaction between different H1 molecules. So this forms a structure called solenoid structure having 6 nucleosomes per turn. So this H1 of the one nucleosome can interact with the H1 of the neighboring nucleosomes, resulting in further folding of the fibers. This chromatin fiber, they in the interface nuclei and mitotic chromosomes have a diameter that vary between 200 to 300 nanometer and represents a inactive chromatin. 30 nanometer fiber arises from the folding of the folding um, of nucleosome chains into a solenoid structures having 6 nucleosomes per turn. So what is a solenoid? A solenoid is a coil wound into a tightly packed helix. Solenoid is a type of electromagnet which is used to generate a controlled magnetic field. So this solenoid is nothing but it is a structure having 6 nucleosomes per turn, and this structure is stabilized by interaction between the different H1 molecules. DNA is a solenoid and packed about 40 folds. The hierarchical nature of the chromosomes, we can see this hierarchical nature of the chromosome in this picture. So DNA is a solenoid. So packing of DNA. So far we have discussed how this DNA is packed inside this in this nucleus. So it consists of many parts. Additional set of proteins are required for the packaging of chromosomes at high levels. These proteins are called as non-histone chromosomal proteins, NHC. These proteins are called as non-histone chromosomal proteins. So in a typical nucleus, some regions of the chromatin are loosely packed and they are referred to as a euchromatin. The chromatin that is tightly packed is called as a heterochromatin. If you see the hierarchical nature of the chromosomes, this chromosomes leads to DNA which consists of histones, and this histones leads to the nucleosomes. Nucleosomes are connected in the chromatin fibers, and this chromatin fiber forms a coiled chromatin fiber, and this coil forms a chromatin. Next we move on to DNA replication. Replication of DNA takes place during the S phase of cell cycle. During replication, each DNA molecule gives rise to 2 DNA strands, identical to each other as well as to the parent strand. There are 3 hypotheses of DNA replication. They are conservative replication, dispersive replication, and semi-conservative replication. I repeat, conservative, dispersive, and semi-conservative. In conservative replication, the original double helix serves as a template. Template is nothing but the original or the parent. The original molecule is preserved intact and an entirely new double-stranded molecule is synthesized. In conservative, the original is preserved and they synthesize the double-stranded molecule, new standard molecule. Second one, dispersive. The original molecule is broken into fragments. In this, the original molecule is broken down into fragments, and each fragment serves as a template for the synthesis of the complementary fragments. Finally, 2 new molecules are formed which consist of both old and new fragments. Third one is the semi-conservative replication. This was proposed by Watson and Crick in the year 1953. This mechanism of replication is based on the DNA model. They suggested that the 2 polynucleotide strands of DNA molecule unwind period and start separating at one end. During this process, covalent hydrogen bonds are broken down. So the bonds between these bases, adenine, thymine, cytosine, guanine, so it breaks. They separated single strand, then act as a template for the synthesis of a new strand. You can see in this picture. Subsequently, each daughter double helix carries 1 polynucleotide strand from the parent molecule that act as a template, and the other strand is newly synthesized and complementary to the parent strand. Semi-conservative replication was proposed by Watson and Crick in the year 1953. The mode of DNA replication was determined in 1958 by Millison and Stahl. They designed an experiment to distinguish between semi-conservative, conservative, and dispersive replication. So in this, they find the parent DNA and the new daughter DNA. The DNA strands divide into the parent DNA and the new strands will be formed. So you must distinguish the parent DNA and the new daughter DNA. In their experiments, they grew two cultures of E. coli for many generations in separate media. The heavy culture was grown in a medium in which the nitrogen source NH4Cl ammonium chloride contained the heavy isotope N15, and the light culture was grown in a medium in which the nitrogen source contained the isotope N14 for many generations. So the heavy DNA could be distinguished from the light DNA with a technique called cesium chloride CSCl or density gradient centrifugation. In this process, heavy and the light DNA extracted from cells in the two cultures settle down into two distinct and separate bands. The heavy culture was then transferred into a medium that had only NH4Cl ammonium chloride and took samples at various definite time intervals, that means 20 minutes duration. After the first replication, they extracted DNA and subjected it to density gradient centrifugation. The DNA settled into a band that was intermediate in position between the previously determined heavy and light bands. After the second replication, that means 40 minutes of duration, they again extracted the DNA samples and this time found the DNA settling into two bands, one at the light band position and other at the intermediate position. These results confirm Watson and Crick semi-conservative replication hypothesis in this density gradient centrifugation of DNA substances with help of nitrogen source. Next we see the enzymes and the mechanism of replication. In prokaryotes, replication process requires three types of DNA polymerases, that is DNA polymerase 1, 2 and 3. DNA polymerase 3 is the main enzyme involved in DNA replication. DNA polymerase 1 is also known as Kornberg enzyme and DNA polymerase 2 are involved in DNA repair mechanism. So, DNA polymerase 3, it is involved in DNA replication, whereas 1 and 2 are involved in repair mechanism. Eukaryotes have 5 types of DNA polymerases that catalyzes the polymerization of nucleotide at 3-OH of the new strand within a short period of time. E. coli that has 4.6 into 10 to the power of 6 base pair completes its replication process within 38 minutes. So the base pair in E. coli is 4.6 into 10 to the power of 6 base pair. So it completes its replication within how many minutes? 38 minutes. Replication takes place faster at the same time accurately. Any error will lead to mutation. However, replication errors can be corrected by repair enzymes such as nucleases. Nucleases is a repair enzyme. Deoxynucleotide triphosphate act as a substrate and also provides energy for polymerization reaction. So for polymerization reaction, one enzyme called deoxynucleotide triphosphate, it provides energy. Replication begins at the initiation site called the site of origin of replication. In prokaryotes, there is only one origin of replication, whereas in eukaryotes with large DNA molecules, there can be several origins of replication that is called as replicons. Since the two strands of DNA cannot be separated throughout the time, that means due to large requirement of energy, the replication occurs within a small opening of the DNA helix called replication fork. What is called replication fork? Replication fork is nothing but a small opening that find at the at the time of replication in a DNA helix. Unwinding of DNA strand is carried out by DNA helicase. Unwinding DNA பிரிச்சு விடுறது. Unwinding of the DNA strand is carried out by DNA helicase. Thus, in one strand, that means the template strand with polarity 3' to 5', the replication is continuous and is known as leading strand. This template strand is called as a leading strand, while in the other hand, the coding with the polarity 5' to 3', replication is discontinuous, known as the lagging strand. So what is leading strand and what is lagging strand? Leading strand is continuous with 3' to 5' end, and lagging strand is discontinuous with 5' to 3' end. The discontinuously synthesized fragments of the lagging strand are called Okazaki fragments. So what is meant by Okazaki fragment? It will be asked in 2 marks. The discontinuously synthesized fragment of the lagging strand is called as Okazaki fragments, which are joined by the enzyme called DNA ligases. DNA can be joined by an enzyme called DNA ligases. As they move away in both directions, newly synthesized complementary nucleotides are paired with the existing nucleotide on the parent strand and covalently bonded together by DNA polymerase. So, this template strand it pairs with the complementary strand by DNA polymerase. Formation of new strand requires a primer. Such the primer, we can use RNA as a primer for the initiation. The primer produces a 3' OH end on the sequence of ribonucleotides to which deoxyribonucleotides are added. So, one primer is needed to join two DNA strands. That primer will be the RNA. The RNA primer is ultimately removed, leaving a gap in the newly synthesized DNA strand. It is removed from the 5' end one by one by the exonuclease activity of the DNA polymerase. After forming a double-stranded structure, this 5' end RNA will be removed. Finally, when all the nucleotides are in position, the gaps are sealed by the enzymes called DNA ligases. Here, DNA polymerase and DNA ligase plays a very important role in the replication process. So, at the point of origin of replication, the helicases and topoisomerases, that means it is also called DNA gyrase, unwind and pull apart the strands, forming the Y-shaped structure called the replication fork. You can see in this picture the replication fork clearly. There are 2 replication forks at each origin. The 2 strands of DNA helix have an antiparallel orientation. They run antiparallelly. The enzyme DNA polymerase can only catalyze the addition of a nucleotide to the new strands in the 5' to the 3' direction, as it can only add nucleotide to the 3' carbon position. Students, I hope you all can understand the properties of genetic material and how the DNA is present inside the nucleus and what is DNA replication and the enzymes and mechanisms of replication in detail. If you have any doubt, feel free to ask me. Till then, safe at home. Thank you.